My name is Haley Bates, and this is a comparison of learning motivation between visual and somatosensory stimuli in Danio rerio. Danio rerio are commonly known as zebrafish. They are extremely popular in research as of late because of their ability to learn via operant and classical conditioning. Cognition is an essential survival skill for the species due to the fact that they are highly gregarious, relying on social hierarchies and shoaling. This means that they need to be able to recognize members of their own species versus predators, which for this study I focused on the Indian leaffish. However, they are preyed upon by just about anything that can get its mouth on them. It's also important for them to be able to remember areas of their environment that are more threatening than others, safe areas, as well as food sources. In a lab setting, multiple different types of stimuli can be used in order to elicit learning. Some popular ones include shock, food, predator images, social rewards, and drugs. However, the possibilities are essentially endless. So this leads to the question, due to the wide range of stimuli available, which stimuli are going to produce a better learning response than others? And is there any difference in learning response? So does the type of stimuli presented to Danio rerio impact learning? And my hypothesis is exposure to a somatosensory stimulus may result in increased learning motivation of Danio rerio in comparison to that of a visual stimuli. So in this instance, somatosensory refers to a physical sensation experienced by the zebrafish, and visual stimuli is a moving sprite image that hopefully the zebrafish are able to discriminate as friend or foe. So in order to run these behavioral trials, I used a Xantex unit. This is an automated behavioral device that allows researchers to code and design their own behavioral trials. And the automation aspect cuts out room for human error in the administration of stimuli, so it makes things a lot more consistent. So the behavioral trial that I ran was condition place preference. This is used to measure a test subject's ability to demonstrate associative learning abilities as well as memory retention over time. So this is just a little bit what CPP would look like. So you would begin with equal areas of each color, and there's no stimulus in this baseline trial. So you're just measuring the percent of time spent in each zone, and whichever zone has the higher percentage is the originally preferred zone. So in this case, it's the blue. And then we're going to condition the fish against blue in order to attempt to flip that preference to red. So during conditioning, the administration of a positive punishment of shock is administered to the blue area, which creates an aversion to the blue. And if learning occurs during your probe trial, which is the exact same as your baseline, you're going to see a flip in the preference to red. CPP can also be conducted using a positive reinforcement. So in this one, the fish originally preferred red. We're going to try to flip that preference to blue using a food reward. And if learning occurs, then the fish is going to associate the color blue with the food reward and spend more time in it during the probe. So for my experiment, I did 5 minutes baseline presence testing on day 1, followed by conditioning, another round of conditioning on day 2, and on day 3, there was a 5-minute probe. So for my experiment, there are 3 different groups, each containing 10 zebrafish of the PetSmart F6 strain. Group A was a social reward, which is the visual stimuli. So it's an image of a zebrafish school moving at different speeds in different directions in their least preferred zone for 2 minutes. 2 minutes on each color flipped 3 times. Group B was a predator punishment, which is also a visual stimuli. So it's an image of a predator, the Indian leaffish, moving around in their preferred zone for 2 minutes to try creating a version for that color. Same time, same amount of flips. And then group C is the somatosensory shock. So the preferred color is flashed directly before the administration of a shock to try to create an aversion that color, and then 2 minutes on the least preferred side, flipped 3 times. So each group had stimuli shown 3 times during each conditioning trial group. So this is the result of a paired t-test. It's the average proportion of time spent in an innately preferred zone before and after conditioning, with this axis representing the percent of time spent, and this axis representing the stimulus and its respective p-value. So ideally, if learning occurred, we want to see a high blue bar, so a high baseline preference, followed by a low orange bar, which is the probe percent time spent in the originally preferred zone. So that would show that they flipped their preference. And then these bars are representing plus or minus 1 standard deviation. As you can see, that's not what we saw for social. We're approaching significance with shock, as we want this p-value to be 0.05 or lower to be statistically significant. So we're approaching significance here, and we had the opposite of what we expected to happen in the social because the orange bar is higher than the blue bar. And then for the ANOVA test, the standard deviation is plus or minus 1 with the error bars. And this axis is percent change. This axis is stimulus. And we're looking at average change in time spent in an innately preferred zone after conditioning. So if learning occurs, we want to see a high percent change, a high positive percent change. And as we can see, we see the social with negative and a not statistically significant p-value. This is the same graph, but with the addition of the post hoc results. Once again, no statistical significance, but there is marginal significance in this one between social and shock. Is this value here? So in conclusion, my hypothesis, exposure to a somatosensory stimulus may result in increased learning motivation of Danio rerio in comparison to that of a visual stimuli, is not statistically supported, but it is marginally supported by the data due to the fact that the shock group showed a marginally increased learning response than that of the social group. I feel an increase of the group sizes and trial times may produce more statistically significant results because of the time constraints. I had to keep the group sizes pretty small and the trial times pretty small. So an increase may allow the fish to really build up that associative learning. However, I do feel that in the social reward group, they may not have recognized the sprite images as a social reward, and it may have been more of a fear response than a positive reinforcement. It's important for researchers to keep in mind that different forms of stimuli at once produce different biological responses and urgency in zebrafish, and honestly, any animal. And because of these differences in urgency and biological responses, there will be differences in learning motivation. The presence of a predator in your immediate space is going to be a lot more urgent than maybe missing a meal. So it's important when designing our experiments to keep these things in mind, including individual differences like sex, strain, housing, and natural history. And I'd like to acknowledge Levi Storks for his help on this project, the Wong lab members, Young Nebraska scientists for the opportunity, and our funders. Thank you.